Richard Hodes, the Director of the National Institute on Aging. It's my pleasure to welcome you to this special Tuesday WALS lecture, 3 formerly blind mice reprogramming tissues to be young again. So I'm honored now to introduce Dr. David Sinclair to you. He's the Professor of Genetics at the Blavatnik Institute and co-director of the Paul Glenn Center for Biology of Aging Research at Harvard. Dr. Sinclair is focused on understanding why we age, and with that understanding, to make us age better. His team recently found that it's possible to epigenetically reprogram cells to safely reverse aging in vivo and regain eyesight. This was published work supported by NIA and the National Eye Institute. Came out in Nature a short time ago. David had his early training and education through PhD in Sydney, Australia. His postdoctoral research then was in the laboratory of Len Guarente at MIT, where they co-discovered some of the early changes that relate to aging in yeast and carried those through to translation through other species. He's been at Harvard Medical School now since 1999. David is also co-founder of several biotechnology companies, serves on boards of others. He's co-chief editor of the scientific journal Aging, and received a number of prestigious awards, just among them, the Australian Medical Research Medal, the Irving Wright Award, the NIH Director's Pioneer Award. Time Magazine has recognized him as the list of top 50 people in healthcare and the 100 most influential people in the world. And he is also an officer of the Order of Australia, which may be a first for our WALS lecture series. Without taking further time, please join me with a warm welcome for David Sinclair. David.
Yeah, thank you, Dr. Hodes. Thanks, Richard, for the kind introduction and the invitation to give a speech today. It's really it's great to be here virtually. Yeah, thanks, Kim and Karen, for organizing this too, and Dr. Gottesman, good to have you here. Yeah, so today I wanted to talk about new work that's come out and coming out from my lab regarding the role of not so much genetic change, but epigenetic change in the aging process, if you want to call it a process. We'll get started. There are some questions that I think are important. Actually, before I do that, it's remiss of me to not thank the NIA in particular for supporting my lab for the last 20 years. I don't often get a chance to directly thank folks at the NIH. And I've collaborated with many people down there. So yeah, I just wanted to really, express a heartfelt thank you for all the support that made my career and this work possible. So what we like to ask in my lab is some big questions. What is aging? Why do we change over time? We take it for granted, but we still don't have a good idea what's driving this process. We know a lot of the hallmarks of aging, many of which you've probably heard of: mitochondrial dysfunction, stem cell loss, telomere shortening. There's a list of 8 or 9, depending on who you ask. But one question that we have in my lab is, how many of those things are tied together, and is there anything upstream? Is there a source of water for the stream, as it were? That, I think, I'll present some evidence to at least get that conversation going. Can we quantify aging? And actually, over the last 10 years, the field's been very successful, increasingly so, at quantifying the aging process, not just qualitative measurements. Can we slow down aging? Now, the reasons for doing that, I think, should be self-evident, that aging underlies most age-related diseases. It's a large component of why we succumb to these very common diseases, from Alzheimer's through to heart disease and diabetes. And if we could understand what's at the basis of that, we may have a way of not just delaying these very expensive and damaging diseases in society, but perhaps even reversing those. And on that note, one of the things that we've been working on is once we think we have a handle on what's driving aging, can we try to reverse aspects of it and quantify that to really show that we're making a difference? So this is a bit of a roadmap to tell you where we're going in this seminar and where we're not going. If you've ever heard of my lab, you'll know that we've worked up here for a while, looking at the role of the sirtuins, which are epigenetic regulators. These will be part of the story, but not the main focus today. And you've probably heard of the drugs metformin and rapamycin. Basically, this has been a lot of work in the aging field for the last 20 years, leading to the paradigm that the environment and molecules that stimulate, or I should say simulate, fasting and exercise can provide health benefits, if not an increased health span and longevity. You've probably also heard of Synalytics, which are molecules being developed in animals and being tested in humans. No positive results that I'm aware of yet, but these are molecules that can either kill or modulate the gene expression of what are often known as zombie cells, senescent cells in tissues that drive up inflammation and may also accelerate aging. And this is addressing what I'm calling on this slide the A3 stage, the very end of the life of a cell. We don't know if you can reverse this easily in vivo, though we are trying that. But what I want to focus on today is an area that's been really at the core of my career, really since I was at MIT in Lenny Guarenti's lab, and that is the role of gene expression, what we're now calling the epigenetic basis of aging. And what my lab and our colleagues and collaborators have recently worked on is it possible to take a cell that is old based on a variety of measures, one of them being a loss of gene expression, the other being loss of function, and can we get them to go back not all the way to being embryonic, which would be the quickest way to getting cancer in vivo, but can we partially reprogram a cell to restore a healthy, youthful gene expression pattern permanently, or at least stably, without going too far? And that's what I want to talk about, early work in that regard today. So this is an egg cell, and when it's fertilized and you wait a little bit, shake it up, cook it up, and you end up with one or more of these if you're lucky. And this is really quite a remarkable process. I think I don't need to tell you that, but trying to understand how this happens, you have to understand epigenetics. How is it that cells with very similar genomes are able to produce cells with all of these varieties of functions and maintain those functions and cell identities for up to 120 or so years in some very lucky individuals? How does that happen? And fortunately, we have increasingly better tools to understand how this actually happens. In these babies, actually, there's 26 billion cells. Each one of them has to be stably maintained. And if you get a rogue cell, you get cancer. If it becomes dysfunctional, you get disease, or at least a bunch of cells and tissues. And one of the first people that got a lot of credit for trying to understand in concept how this happens from a fertilized cell or a stem cell to a tissue is Conrad Waddington, who in the 1950s proposed what you may have heard of as the Waddington Hypothesis, which is the idea that cells that have no identity will metaphorically roll down a hill and end up in valleys where they stay, so that a skin cell doesn't suddenly turn into a liver cell and a kidney cell doesn't turn into an eye cell. Of course, there's many cells within those tissues as well that needs to be maintained. But in the aging field, we very rarely, if ever, thought about development, and vice versa. Those studying development had really little interest in aging. But that's changing. Many people in the aging field have now thought about this, at least, that perhaps aging may not be a evolutionarily conserved process to cause aging. We don't believe aging is an adaptive event, but it may be related to the epigenome. And what I mean by that is, and there's increasing evidence across the field, some from my lab over the last decade, is that cells that are terminally differentiated can actually move around on this Waddington landscape and start to become more like other cell types over time. And we see this happen during aging. We don't know if it causes aging, but it certainly doesn't help their gene expression patterns. And in my lab, we call this X differentiation. You can call it what you want. Could be epigenetic drift, epigenetic noise, but it definitely happens when you look. And we have tools now to look at this at the single cell level. So that's pretty well established. But the question is, could you ever get the cells to go either back up the hill and go back to where they came from, or ever go back all the way up the hill? Well, we know from Shinya Yamanaka's work that you can go all the way back up the hill, but can you go sideways? That's been the question. So if you're very young, you probably only have heard of what these things are. These are compact discs. We used to store movies and photos on them. They were pretty exciting at the time. And what I want to use this image as an analogy for a cell. Now, there are two types of information in a cell. Predominantly, they're stored in the genome. This is a digital code, much like the pits in this CD. And it's, instead of being binary, it's base 4. You got A, T, C, G. But in our bodies and in a CD player, there's analog. You still have to convert this to sound. The reader is still physically moving around to find the song. Similar to the epigenome of our cells, much of that is analog. And anyone who's dealt with an analog device, whether it's a record or a tape, knows A, that they don't last that long, and B, they're very bad at being copied. And you introduce noise over time, similar to Xeroxing something many times. And I think it'll be a useful analogy later. Another concept that I want to introduce, if you're not familiar with it, is the concept of information transmission and storage. And this fellow up here was a professor, Claude Shannon. He was at MIT during the 1940s and spent much of his career trying to figure out how to transmit radio signals without it being lost along the way, which of course can cost lives, as they did in World War II. So he came up with a mathematical theory of communication, and it basically led to the internet that we use today. And what he said in this diagram, this is pulled straight out of his, at least the black part is pulled straight out of his paper from 1948. He said that you can have a transmitter and a receiver, and there will always be noise, whether it's background radiation or something. But the problem is, how do you have a perfect transmission? And he proposed that there would be this observer. We now call the observer the backup copy or the TCP/IP protocol of the internet. But he was the first to show, mathematically at least, that you could use a backup copy to reconstitute the original message. And this is why we can no longer use the excuse that, oh, I didn't get your email. It always arrives, unfortunately. But what I've done is I've added a concept of biology as I'm thinking about it currently. And that is that information, either genetic or epigenetic, is in a relatively pristine state when we're very young. And over time, you have aging. And what might be happening is that you lose information, whether it's genetic or epigenetic. And we don't know what that is yet, for sure, especially the epigenetic component. And perhaps, is there in cells a reset switch? Did cells ever store a more youthful version of their gene expression pattern, say, or their younger identity? And if they do, wouldn't it be amazing? Because then we could potentially tap into that. And as I show in this example of the eye, reboot the system and get those cells to function like they were young again. So this is where we've been headed lately. For those of you who don't think about epigenetics all day, just very briefly, an update. Gene expression is controlled by closed chromatin or open chromatin. There are big loops, there are small loops, there are contacts. We can now read all of these in 3 dimensions using technologies that are recent. We've got modifications of the histones that package the DNA, methylation, ubiquitination, there's acetylation. One of the main modifications that I want you to remember is DNA methylation. This turns out to be a very good marker of aging cells, but also increasingly thought to play a role potentially in this information storage that I'm referring to. And we can measure all this, and I'll show you some examples of that. We can use it to look at the aging process as well as the actual estimated age of a tissue. This is a little complex, but all you really need to know is that there are enzymes that control DNA methylation, the chemicals that are added and subtracted to DNA to control it. This is basically the DNA version of the compact disc player. And they decide, or they tell the cell in part, which songs to play when, and which cells should become skin cells and which cells should stay brain cells. And some of the genes make TET enzymes. There are a few TET enzymes, and they, in this case, they will remove the methyl tags. And there's another one called TDG that we've worked on. And this cycle controls the on and off process, but mostly it's very stable, as I mentioned, because you don't want your eye suddenly behaving like a liver, just to put it as an exaggerated example. A real breakthrough in the field came from Stephen Horvath's lab and Greg Hannum's. They get the credit for showing mathematically that when you measure the methylation sites across the genome and plug those changes that occur during aging, whether it's passage of cells in a dish or passage of time in a human, you actually get a predictable straight line. What's interesting, though, is you can see that even during development, there are changes in these chemical patterns, the methylation pattern, which Steve likes to joke or perhaps argue in a mathematical sense that we start aging at fertilization. But what's important is that after the teen years, you can draw a straight line and use machine learning to train this. And you come up with a set of sites that when they go up or down in a certain way, you can then predict what we call the biological age estimation of a cell or a tissue. And so I could use this technology to take your blood sample, spin it down, get the white blood cells out, and give you an estimate of your biological age, which might actually be a few years different, up to 10 years different, I believe, than your actual birthday candles. And increasingly, the field has shown that people who have a higher biological age are more susceptible to diseases of aging. So that there does seem to be some validity to these changes. But whether or not these changes have anything to do with causing disease, besides cancer, which has been known for many years to be related to this, but other diseases, do we know if it drives aging or is just a clock on the wall? We don't know that yet. So back to this analogy, these CDs in this analogy get scratched. So a scratch CD will skip and the reader will not read the right songs. And that's a problem. And it's similar to what happens in a mammalian cell. And in basically any eukaryotic cell, they don't read the right genes at the right time. And it just gets worse and worse over time till eventually that you just switch the machine off. You don't want to hear it anymore. And if I'm talking about cells, the cells we can see have lost their youthful gene expression patterns and actually look like they're starting to express genes that should really have no place within that cell type, which I'll show you evidence of in some coming slides. But imagine if you could polish this CD and get it to play the beautiful music again. That's what we're trying to do here, if it's possible. So a little bit more representation of what I'm talking about. This is the chromosomes in a cell. This could be a yeast cell, could be a mammalian cell. They're shown in black. And these are areas of heterochromatin, which silence genes. Some of them are found at the periphery, such as the sirtuins that I've been working on for most of my career. They control silencing and heterochromatin. And there are open genes that transcribe RNA, and those genes are on and often in loops that are exposed to the cell machinery. But over time, what we see is changes in those, and genes that should be left on become switched off and vice versa. And we believe this is leading to a partial loss of identity. But the question is, why would this happen? How does it happen? So we can do two tests. One is, and what we've actually done, is test some cellular perturbations. We've tried DNA damage and we've tried cell injury and asked, does that mimic the natural changes during aging? So what is the source of the noise? Now, one big clue came in the early 1990s. Now, Brian Kennedy, Nick Austriaco deserve a lot of credit. In 1994, just as I was coming to the US from Sydney, Australia, they put out a paper in Cell that said that the SIR4 gene, SIR standing for Silent Information Regulator, information is important in that acronym, that mutant in a yeast cell was able to extend lifespan. And what then we showed with Brian, and published a paper shortly thereafter when I got to the lab, was that the reason you get this protection from aging and longer lifespan is associated with and because there's a relocalization of the SIR protein complex away from silent loci, such as these mating type loci and telomeres. These are heterochromatic, and they move to a site of genome instability, which in yeast and in our cells, but in yeast we studied it, it's the ribosomal DNA. And the ribosomal DNA is very repetitive, about 150 copies end on head to tail. And we could see them move there. And we could mimic this effect by just breaking the chromosome really anywhere we wanted to. And we saw most of the SIRS complex move to the break. And that included the protein called SIR2, which gave rise to the family of sirtuin proteins. Now, sirtuins, I'm not going to talk about much more again. I just want to tell you that they control heterochromatin, we know, because they take NAD as a co-substrate molecule, which is changing levels during aging and in response to what animals eat. And then they deacetylate histones to compact that chromatin. We also had a look at what happens during aging in a mammalian cell. Is there something like this going on? In other words, do sirtuins, which have very specific places at about 400 silent genes and repetitive DNAs in mammals, including transposons as well, retrotransposon, do they move during aging? And can we promote their movement by creating a DNA double-stranded break? And it's actually known that chromatin reorganizes in a quite dramatic way in response to double-stranded breaks. But what isn't known is what happens to the rest of the genome. Does it affect heterochromatin? Does it affect the epigenome? And are there permanent changes after the repair has happened, even if there's no mutation left behind? And so this is the concept that in a young cell, you have this pattern of loops and bundles that control gene expression. If we create a break in the chromosome of a mammal, will we get relocalization of those proteins to the break? Yeast says yes. We had to test it. And then over time, if you repeat this, many hundreds of times, and consider that in our 26 trillion cells in our body, this happens at least once per day. So that's a lot of breakage over time. Do we get this disruption? So we could test that in cells and we could test this in mice, which we did. And this was a review that came out. It was in Cell by Jan Vij, if you say correctly. And what he was describing was a study where we created breaks in mammalian culture, mammalian cells, and in mice. And we found that the relocalization required these DNA damage checkpoint proteins. So this wasn't just a random drift. This really was an active process that required phosphorylation, chemical modification of the actual histones at the break. And it's one of the first steps, the movement of SIRT1, which is the homologue of the SIR2 yeast gene that I talked about. This enzyme gene product, the enzyme moves to the breaks, recruits repair proteins, and then most of it goes back to where it came from. And we could see this also recapitulated during aging, this process of moving away from the SIRT1 regulated promoters. This enzyme was very useful to us. It's been used to study DNA break repair for many years in vitro, but we started to use it in vivo. We use it in our lab because it doesn't cut very often. There's only about 18 consensus sites across the genome. It does cut the rDNA, which we had to be very careful. We weren't just trashing the genome, but it's been very useful because it stimulates a very, very mild damage response. In fact, it's so mild, you can barely detect any activation of these factors. By a Western blot, you won't see P53 activated, for example. We call it chromosome tickling to distinguish it from other types of study. So now we could ask the question, in cells and in mice, does the response to DNA damage, in particular to double-strand breaks, which are very deleterious to a cell, does that induce temporary or permanent changes in chromatin, and do they potentially drive aging like they do in yeast? So we built these mice. We engineered these mice called ice mice, which Philip Oberdorfer, who's now at the NCI, created in my lab about 10 years ago. He's been using them too. And we call them ice mice, which stands for inducible changes in epigenetics. They have nothing to do with border patrol. And we have MEFs, which are embryonic fibroblasts, and adult fibroblasts that we can take from these mice. And this is work mainly by Jae-Hyun Yang in the lab, a talented postdoc. And he has developed this in vitro system with fibroblasts from these mice where we can induce this cutting and let it heal after 24 hours. This is tamoxifen inducible, and it's very tight. And afterwards, the cells just are fine. You cannot tell the difference between these cells and the original cells if you just look at them. And actually, I should mention, they don't even slow down in the cell cycle. It's very mild. Now, if these go on further and you leave them a few weeks, they will have more cellular senescence. So we think we're inducing a very, very early pre-senescent state. But we could ask the question, are there any mutations, changes in cell cycle, which I've said there aren't, loss of epigenetic information, and is there noise? Now, I'm not going to show you the data. It's actually on bioarchives if you want to check it all out. We spent about 4 years looking for mutations in these cells and in the mice that I'm going to show you. We could not find any increase in changes in mutations, and we looked carefully at the cut sites and across the whole genome. So we don't think any changes that we're seeing are due to mutations. We also don't see any epigenetic changes specifically at the cut sites, at the repaired sites after recovery. And we can use a number of very sensitive methods to do that. In this case, this one's called HiChip, that looks at a number of proteins and modifications at this site. So this is a two-dimensional version of three-dimensional associations of the genome, and nothing's here. If there were changes, you'd see differences between this lane and this lane here, and this lane versus this lane. The reason it says CRE is this is a recombinase that induces the cutting enzyme. And here's the ice. All right. So I will show you later what happens in 3 dimensions, but I want to mention first that when we look at changes in gene expression in the cells that have been cut and repaired, what we see is an abundance of changes in genes that are involved in cellular identity and cellular development, embryonic development. You can see, hopefully, if you can read these words, there's a lot of morphogenesis development there. And when we do the overlap of these changes at the histone level, there's really a striking overlap between gene sets that fall into this class called the Hox genes. So what are the Hox genes? Well, you might remember from biology that they control where your head is and your tail is. Okay, you do not want these disrupted, but here we have these disrupted after just 24 hours of DNA repair. And when we look across this locus, we've done the whole genome, but I'm just showing you this locus. The control is in blue, looking at the acetylation levels of histone 3, K27, lysine 27. And there are definitely changes going on here. Shown here, there's less acetylation in the blue, and over here, there's more acetylation. And this is across a very large part of the genome that encompasses 13 different genes. And correspondingly, there's changes in gene expression as well. These genes get switched down, and these genes get switched off. And this is an epigenetic change because it survives multiple passages going forward. Now, this is the three-dimensional structure. These red lines show the interactions. And you can see that there are big changes after the repair of this break, such that there are new enhancer elements, as they're called, that are now interacting with these genes down here, this cluster. And so I can say with some certainty that even if you repair a broken chromosome and there's no mutation left behind, there are epigenetic scars left behind. And potentially this could drive, in part, the gene expression changes during aging. But what about if you do this to a mouse? What happens if you scratch the CD? And so these ice mice we induce, not for 24 hours, but we do 3 weeks of induction using tamoxifen when the mice are 5 months old. And then we let them, we turn it off, and then we let them go to see what happens. Now, what's important is that during the treatment of 3 weeks, there's no big DNA damage response. The mice are healthy. In fact, they don't respond to anything. It's like getting an X-ray, presumably. And 10 months later, this is what the control mice look like. And we've assessed now many hundreds of mice. So it's a very clear phenotype. This is the control. So these are now 15, 16 months old. And the one that was treated earlier started to develop a phenotype that ended like this. This is what these ice mice look like. And I'll show you a little bit of data, but we've got 10 years of data on these mice. When we hand out these organs to the aficionados, whether it's the skin or the eye or kidney, they're pretty surprised at what a good, they're surprised how good a mimic this is of aging itself at the histological and gene expression level. And I can tell you that they have defects in memory, they have defects in endurance, they've got kidney defects, heart issues, their skin is obviously looking old even up close. They develop vision issues, and you can see they have alopecia and gray hair. You could even argue that they're even more aged looking than a normal black 6. This is a black 6 mouse here. We can actually dial up the aging amount if we want. If we give them less tamoxifen early in life, they don't age, quote unquote, age like this. They actually are less. Okay. So is this really aging? Well, it's hard to tell really, but you can try. They have the hallmarks of aging. We've looked at their mitochondrial loss. We see that. And this is just a compendium of a lot of that. They have more frailty. You can see here. They have osteoporosis. It goes on. The metabolism looks old. Their brain, we can turn it on in the brain. We can basically just, let's look down here. They have defects in, over here, they have defects in memory, which are similar to old black 6 mice. Same with the Barnes mice. So, you know, I could go on for another 20 years showing you that they resemble aging, and you still wouldn't believe me. But fortunately, you know, Steve Horvath and Hannum came along and they said, well, you don't have to do this qualitatively. You can have a look at the DNA methylation clock. And so we did that. And these mice, based on the methylation clock, are 50% older on average, which was nice. I mean, that is the first evidence that we can accelerate the epigenetic clock through some mechanism. You know, we still have a lot to do to figure out how a DNA break leads to this, and we have our hypotheses. But this at least gave us comfort and reviewers comfort that this is resembling aging, if not a mimic of it. But the question now is, can you reverse that? And so this is where reprogramming comes in. I like to show this slide mostly to the lay public because it gives you an idea of what the cell has to really do to be reprogrammed. Those beautiful loops and bundles of chromatin have to somehow get back into their original organization. And that's not a simple thing. It's not even clear if that's doable. So let me show you that it might be doable. Now, one of the clues that it might be possible is the following. Thanks to John Gordon's work with cloning of tadpoles, somatic nuclear transfer, we've known since the 1970s that you can take the nucleus of a tadpole and regenerate another tadpole. Shinya Yamanaka in his lab showed that you can take an adult mammalian cell and using 4 genes listed here, Yamanaka factors, you can have induced pluripotent stem cells. So together, those two professors won the Nobel Prize in 2016, deservedly. But that doesn't show that you can improve the health of a living animal without causing it to either become a giant tumor or having to go through a single cell stage. But we have some clues. We do know some species can regenerate themselves dramatically and, quote unquote, be immortal. It's hard to prove immortality, but it's pretty close. You can even take off the limb of some species such as axolotls, even lizards, of course, regrow parts, and young mice can regrow fingertips. So this is not an impossibility. In fact, in humans, you can take out a chunk of liver and it regrows surprisingly quickly, as long as you're still youthful. And so that we think, at least in my lab, this is a drawing from a review that we're putting together, that these processes may be linked somehow. We have spent a lot of time recently focusing on neurons. We find them a very useful model for rejuvenation for the following reason. A very young nerve cell will regenerate. It will grow in the dish. If you break the spinal cord of a very young animal, it starts to regrow back. But very soon after birth, mammals lose this ability. And if you take nerves, from the central nervous system and plate them, they're not going to grow back. Similarly, if you damage your optic nerve, it won't grow back. And spinal injury is a permanent problem for those who are unlucky enough to experience it. But imagine if you could take an adult nerve cell in the spine or in the optic nerve or in the brain and send it back far enough that it had this property, but did not lose its identity. That's the goal here. And so we took a leaf out of 2 labs. The first was the Yamanaka Lab, where we thought perhaps reprogramming cells might be possible. Of course, we do not want to go so far that we create stem cells in the body for obvious reasons. And in fact, it's known that if you induce all 4 of these genes in a mouse transgenic strain, you will kill the mouse. And if it survives, it will develop cancer. So this is not a very safe way to go. And it's not going to be a therapy anytime soon. But it's still pretty interesting that it's even possible to do this at some level. Now, another breakthrough came, this time by the postdoc Alex Alcampo and in the Belmonte lab. This is Juan Carlos Belmonte at the Salk Institute. In 2016, you may be familiar with their cell paper. They had a progeroid model, which is the laminae progeroid model that I think many of you at the NIH know, Hutchinson-Gilford syndrome, progeria. And this mouse model gets very sick very quickly in a matter of months. And what the Belmonte lab did, which was very clever, was to turn these 4 genes on for only 2 days and then let that mouse recover for another 5 days by switching them off again. And they found that the mice didn't die or develop a lot of tumors. And they could now ask the question, are those prematurely progeroid mice better off? And they were. They lived about 30 to 40% longer and had some improved health as well, which was remarkable. But it didn't really tell you if it was safe. It didn't tell you if it was relevant to a normal mouse. And it didn't really tell you that age was going backwards. I'll be honest, some of my colleagues, not me, but there was some skepticism that you were just stressing the mouse out so much that it was a stress response, not really reprogramming. But we thought that it had a lot of legs, and we were already trying to figure out combinations of these genes that might be safe. And so a talented student at the time, who's now a postdoc in my lab, moving actually to Jonathan Weissman's lab next month, and the first author of the paper that we just put out, Wancheng Lu deserves a lot of credit because he was almost ready to give up because he had been trying for 2 years to find a way to partially reprogram a cell to be younger, but not strip so many DNA methyls off the cell that it would be pluripotent. Unfortunately, he didn't give up. What he developed as his last-ditch effort was a controllable system in an adeno-associated virus, or AAV, where he could express just 3 of the Yamanaka factors, and he left off the c-Myc gene, right? This doesn't take a genius, to be honest. c-Myc is a potent oncogene, and why people were expressing it in vivo, I'm not sure, but it was, I think, a fairly straightforward idea to just try these 3 and see what would happen. And he did it. This vector is available to anybody who'd like it. And we have a couple of versions that have higher or lower expression. And you can turn off the system with tetracycline or doxycycline, or you can turn it on in cells or in mice. And we found, to our delight, that if we overexpress these 3 genes in a whole mouse, so we're now using AAV9, which infects many different tissues, particularly the liver, spleen and pancreas, I think the other 2 that were high levels. We didn't see any toxicity. We haven't seen any cancer. In fact, we left those mice for 15 months and we didn't see any toxicity. We don't see induction of nanog, which would be evidence of them losing their identity. So that's good. Cells throughout the body don't seem to lose their identity. And we use the eye as our model in these experiments because remember, the central nervous system is a really great place to test age reversal. And not only that, AAVs are currently used in gene therapy in humans to treat genetic diseases. And so we thought it could be a win-win-win for everybody if it worked. So we initially teamed up with a couple of labs who we still collaborate with. Zhigang He's lab at Children's Hospital at Boston and his lab members studied retinal regeneration from a nerve crush injury. And we work with Bruce Cassander, who works on glaucoma and age-related vision loss. And this is a bigger syringe than we use, but we injected the virus intravitreally, and the virus made its way into the optic nerve cells, which are retinal ganglion cells, or RGCs, and asked the question, if we turn on these 3 genes, do the cells survive injury, or do they regain their youthful functions in the case of glaucoma or aging? And I won't spare you all the details. It's published, but we did a lot of experiments to look at the timing, the dosage, the amount of infection. We can infect about half of the retinal ganglial cells and turn on the 3 genes. And it only worked when we had the 3 genes. If we just did 1 or 2, you didn't get the effects that I'm going to show you. So we were very lucky to pick the right combination, though I'm sure there will be others found further down the line. Ideally, we'd like to do what we're doing with chemicals, and we're screening. But here's the experiment for the nerve crush. It's just a pinch at the back of the eye for the mouse. And then you wait. So what's important is that the OSK for short, OCT4, SOX2, KLA4, it's induced post-crush. This is not a pre-incubation. That's important, obviously, because if there is an injury, you cannot predict when that's going to happen in a human. We can add this dye called CTB and have a look at how far the dye spreads down the optic nerve. So this is no treatment. We're now looking at it in reverse. The eye is on the left and the brain is on the right, off the screen. And there's a lot of neuronal loss here. A lot of these RGCs have died off. This should be bright orange in a normal mouse. And this is where the crush was. And there's no evidence of any neuronal regrowth. Remember, a very young mouse can do this. So in the reprogrammed ones, or the ones treated with OSK, we got a young response. So a lot of the nerves survived and the axons regrew in the right direction. Somehow they know where to go. And if we left this not just for a couple of weeks, we left it for 16 weeks, though many of those nerves, a large amount, actually reach the brain and meet the chiasm where the visual system meets, the optic nerve. And as I mentioned, this only worked, so now we're measuring this distance, this only worked with the 3-gene combination. And we put them in a cistron so they're expressed roughly the same levels. What about the DNA methylation clock? Were these really getting younger, or is it just a, some other phenomenon? So what we found was that, first of all, surprisingly, the injury based on this clock accelerated the age. We didn't expect injury to accelerate age. Remember, we were thinking DNA damage, but now it looks very much like injury is also aging of the epigenome. And what you can also see is that the treated cells with OSK are brought back to a younger level. In fact, the longer we leave this, the younger they get. But if we look genome-wide, which I think is more important than just looking at a few hundred clock sites, it's very clear that the injured ones here with a control vector change their DNA methylome, the pattern of DNA methylation, and the ones that are treated look much more statistically like the ones that were not injured in the first place. And when we do the statistics on how much the injured actually looks like aging, which I'll show you later, the p-value is 10 to the minus 200. So there's a really high and surprising overlap between these changes here during injury and aging itself, an unexpected correlation. So what about glaucoma? Anyone who's had glaucoma in the family will know what I'm talking about. It's a devastating disease largely due to intraocular pressure. And in this mouse model, you increase the pressure in the eye with some beads you put in there. And after 4 weeks, the pressure has all gone, but there's a permanent loss of vision in the mice. Similarly, 70 million people in the world currently suffer from this condition, and there's no way to reverse that vision loss right now. Then we could turn on the treatment for 4 or a total of 8 weeks and ask what happens to vision in these mice. We measure vision in the Cassandra lab a couple of ways. One, we measure electrical activity. What I'm showing you here is what's called the optomotor response, which is mice moving their head if they can see those lines moving left to right. And we can cycle the width of those lines. And I mean, to jump to the chase, you can see that the longer we leave the treatment, the better the vision becomes. So this is the loss of vision and the recovery. And if we don't do anything, that vision never comes back. And strikingly, this is actually data that's better than in our Nature paper. We only did this time point. This is hot off the presses. We'll repeat it, of course. We need to repeat it. But this is the data that we now get at 8 weeks, which is amazing that it's pretty close to the original vision of these mice. Does it require DNA demethylation? Right? Remember those TETs and TDG genes? For the aficionados, TDG is important because that's required for active DNA methylation, enzymatically active, pulling those chemicals off, not just splitting of the cells and diluting the methylation. And so we tested both, and it turns out that if you don't have TET1 or TET2 expressed, you don't get the axon survival. And in the case of the restoration of vision in the old mice, which I'll show you in a second, you don't get that as well. And so it was, I kind of jumped to the punchline, unfortunately. But what you can see here is a quantification of improved vision in regular old mice that requires active DNA demethylation. So this is what it looks like. This is an untreated control mouse at 1 year of age. Now, black 6 mice have a genetic defect that make them lose their vision very young, but they're regularly used for age-associated vision loss. And this mouse is not moving its head, as I hope you can see. It's essentially blind to these lines. But in one that we've now expressed the OSK transgene for a month, I'll give you an example. We see that it's moving its head. Hopefully you can see that mouse is spinning its head to the right. Of course, we've checked electrical activity in the mice, and we can see that that's also improved. So it's not just a camera looking at the mice, it's really physiological. And again, in these aging mice, we can see a lowering of DNA aging signatures And this is the methylation pattern, again, restoration of the DNA methylation pattern to more of a younger DNA methylation pattern. But I think what surprised me even more was the effect on gene expression. Remember, if we're right about this, we should see at least a partial reset of genes that should be on versus off. Now, this is a two-dimensional graph that shows genes that go down with aging and then genes that go back up with treatment. Genes that go up with aging and genes that go up with OSK. So if the treatment accelerated aging or exacerbated aging, we'd see the genes on this axis. And if it reversed aging, it would be something like this, right? Goes down a certain amount and then up the corresponding amount by reprogramming. And this is what we got. That's clearly along the axis that we predicted for resetting of age. This is only the top 464 genes that we've looked at. The red ones are really interesting. Actually, these are all the ones that are involved in, at least believed to be involved in, sensory perception. You can see these are the Go groups shown here. Some of these are involved in smell and taste. Now, your guess is as good as mine what's going on during aging and reprogramming that has to do with sensory proteins. But it's exciting to think that there might be an involvement of chemicals in this process. So we're looking into that. And so I'm just going to summarize now what I've told you and have some questions. What I've shown you is that at least evidence that we can drive the methylation clock, aging clock forward and recapitulate what looks like aging in cells and in a living mammal. We've looked at the three-dimensional structure of chromatin in these cells, and we do see those changes, in particular at developmental genes. And how this happens, we're in the process of trying to dissect that at a very detailed level. We do believe that the sirtuins, at least SIRT1 and SIRT6, are involved in this process. But of course, there are other proteins, there are other modifications that are definitely involved, and we've got a lot of work ahead of us. And it's not just my lab, there's going to be a lot of work required. Same with reversing, quote unquote, reversing aging. I've shown you that injury and aging does change the DNA methylation pattern in ways that are reproducible. Other labs have shown this as well. But what's novel here is that we can reset a lot of these changes, and that requires this active DNA methylation process involving the TETs and TDG. I didn't show you, but based on where these changes have occurred in our system, we implicate the PRC2 complex, which is very well known to control embryonic development. And then I make the case that these cells are younger and seemingly behaving as though they're healthier. And in the case of vision, we can see the restoration of eyesight after the treatment. Now, some of the questions that we want to ask is, how long does this effect really last? Can you go for months or years? We don't know how long we can go. We know it months, but we don't know longer. And how many times can you reset? And how many times do you need to reset? That's also on our list. We've also started reprogramming other parts of the eye. We have some preliminary but really unambiguous results reprogramming the retinal pigment epithelial layer. That's very important for macular degeneration. And we're also working on hearing and have some early evidence that we might be able to improve hearing loss as well, which would be great. Of course, we'll see how many other organs we can tick off. But getting back to Waddington, just to finish up, X differentiation, as we call it, seems to be somewhat reversible without driving cancer and pluripotency. How it actually happens, we're not really sure. We believe that the Yamanaka factors can do this, but how they work and where is that information to reset the cell partially? Is it a DNA modification? Is it a protein? We don't know yet. We're looking for the observer in mammalian systems. it could be a DNA modification that we've never paid any attention to, but we just know it exists. And so that's one of the big questions we'd like to answer. My lab has been awesome, particularly during COVID. They've really done a wonderful job. I think scientists are fantastic when it comes to being dedicated to their careers, and I just can't thank them enough. Many of these people have done the work here, and I tried to credit people along the way. This is Wan Cheng, who's the first author of that paper I was describing. And then here are the lab members that have been fantastic. There are members that have left. A shout out to Mike Bonkowski, who passed away during COVID unexpectedly. Philip Oberdorfer, who started this mammalian project. Big shout out to Paul Glenn, this seed grant that helped us get here. And these grants were instrumental in getting us through the understanding from yeast all the way to the present day, and many collaborators. So I'll leave it here and I'll leave time for questions. Thanks, Richard, and everyone for having me.
Thank you, David. Great talk. Well, let me ask one question. When you were first conceptualizing the state of aging, if you will, at which you were intervening, you emphasized this was somewhere proximal to the potentially irreversible cellular senescence. Have you had occasion to see whether the reversal that you've seen has an effect on the accumulation of senescent cells, if not their actual reversal?
Yeah. Yes, I have a student working on this, and he's got some in vitro evidence, at least, that it does delay cellular senescence. We need to now go into these mice that we've reprogrammed and really look at that. So we don't know yet. I think the hypothesis is valid. It would make sense. Another question that we'd like to answer is, if you reprogram a senescent cell, what happens? Is it bad? Or does it reduce the SASP, the inflammation? Or could you safely restart a senescent cell into the cell cycle? That's really, that would be something unexpected, but we're going to try that.
Let me read a couple of the questions that have come in. Is there a role for inflammation in injury or DNA damage-induced tissue aging? And does inflammation counteract the restorative effects of OSK delivery? Or I guess, does OSK delivery affect inflammation in that direction?
Yeah. We know the first part, that when we age those mice, they get inflammation in the cells, they get inflammatory markers. We see a slight induction of P21, etc. So in the mouse, yes, we've got in the brain, GFAP comes on, the hallmark of inflammation in the brain. That is delayable, if that's a word, by overexpressing SIRT1. That was in Philip's 2008 Cell paper that I referenced. We don't yet know, unfortunately, whether reprogramming will reverse inflammation. We focused on the eye. We'll check that. Based on gene expression, we think that that's going to be true, but we do need to do more work.
Another question that took seriously your damaged disc, I think. Where's the useful epigenetic information stored in the information theory of aging? Where is the backup device?
Yeah, as I mentioned, it's a mystery. I hypothesize that it's probably a DNA modification because that's very stable. And this mark, if it does get put down at the age of, let's say, 5 years old, 10 years old, that needs to survive for many decades. But it could be an RNA DNA hybrid. It could be a protein that binds very strongly. Unfortunately, we don't know. So anyone who wants to try find it, I think it's up for grabs.
Very good. Well, I think the clock says 4 o'clock. I don't know where the count finally stopped, but I know we were over 4 participants while they were seeing us gear up, and probably ended up being more than that. One thing I want to repeat for those people who are interested in CME credit, 31973, and information asking you shall receive. We did hit the 500 mark, and as counting was going on. So a really good turnout, I think, reflecting the interest, and I'm sure the group was not disappointed. So a small group of trainees is going to have the privilege of speaking with you in a so-called tea following. This used to be a walk from the major conference room at NIH down to the library for cookies and stuff, but this is our virtual version of that. So again, the CME code 31973, and Dave, was a fantastic talk. There are many more questions. I'm sure you're open to being contacted directly with those questions as they follow, and we'll have a chance for a number of the trainees to pose them to you in person in about 15 minutes from now. Thank you so much. Great talk.